This is our spirochete lecture, and this lecture has been broken down into 3 different parts. So the spirochetes include 3 different genera. They are Treponema, Borrelia, and Leptospira. Within the Treponema genus, we have Treponema pallidum, which causes the disease syphilis. Within the Borrelia genus, we have Borrelia recurrentis, which causes relapsing fever, and Borrelia burgdorferi, which causes Lyme disease. And within the Leptospira genus, we have Leptospira interrogans, which causes leptospirosis. In general, the spirochetes are very flexible, helical-shaped organisms that have a multi-layered outer envelope, and they have endoflagella attached between the outer membrane and the cylindrical body, which allows their motility through flexion. We're going to start out by talking about the Leptospira organisms. And on the bottom, you can see the spiral shape, which is why they're one of the spirochetes of the Leptospira organism. So Leptospira causes the disease leptospirosis, which is a very acute generalized infectious disease caused by the organism Leptospira interrogans. Now there are more than 200 serovars of Leptospira interrogans that have been reported in the literature. Leptospira is an obligate aerobe, and it's a zoonotic organism that has worldwide distribution. There are multiple types of animals that can carry Leptospira interrogans. So there are many species of Leptospira, but Leptospira interrogans is the most common organism that has been involved in human infection. So the morphology of Leptospira interrogans, it's a tightly coiled, very thin, flexible spirochete, and both ends of the Leptospira organism have little hooks on them. They contain 2 periplasmic flagella. So these are very different flagella than what we've seen with the regular bacteria that we've talked about thus far. So they don't have that projection that's flying out from the bacteria that whips around that allows movement. This flagellum is within the periplasm of the organism, and it flexes and allows the spirochete to flex and move. So it does have a flagellum. It is motile, but it's a very different type of flagellum and a different type of motility than all the other bacteria that we have discussed up to this point. So the Leptospira motion is very rapid, and it's rotational. So there are variable symptoms with leptospirosis, and usually these symptoms are flu-like in nature. And the thing about leptospirosis, it can be confused with dengue viral infection, especially in endemic areas of South America that have both dengue virus and leptospirosis. So initially the symptoms are fever, chills, muscle aches, headache. Can also include vomiting, a rash, diarrhea, and some meningeal symptoms. There is a secondary phase of infection that includes an aseptic meningitis, jaundice, can include kidney failure, as well as hemorrhaging. So the secondary phase can get very severe. So it manifests as a meningitis or liver or kidney disease. Usually leptospirosis is self-limiting, but there are some individuals that get very severe disease. So severe disease, such as Weil's disease, can lead to kidney failure, and there have been fatalities associated with severe leptospirosis infections. But normally, most people will control the infection and overcome it. So, the habitat of leptospirosis is that the organism is maintained in animals, so it has an animal reservoir. You can prevent leptospirosis infection by making sure you drink sterile water, water that hasn't been contaminated with any animal excrement, and also vaccination of domestic animals. So, dogs are vaccinated for leptospirosis because dogs can become infected with it. You can treat leptospirosis infection with penicillin as long as you get the treatment early in the infection. Later in infection, when there's complications and kidney failure, then you would have to start supportive therapy, and antibiotics at that point may not be helpful. So leptospira has a global transmission. You can find it pretty much anywhere. And how humans get infected is through small breaks in the skin or through intact mucosa. The incubation period is 10 to 12 days, but it can range from 3 days all the way up to 30 days. And how individuals normally are infected is indirectly through contaminated urine. And what happens is that animals get infected with leptospira. Animals will then urinate in some type of a water source. And then humans will go into that water source and get indirect transmission from the water that's contaminated with animal urine that now has leptospira in it. So the indirect transmission is the more common way that humans get infected, much more common than directly in contact with contaminated animal urine. So the incidence of leptospirosis varies depending on what region of the world you're talking about. So there are peak incidents of leptospirosis usually in rainy seasons, especially in areas where leptospirosis is more common. So there are areas of South America where during the rainy season and in areas that tend to have a lot of rodents and rats that are infected with leptospirosis and are urinating, the rainy seasons will develop some flooding, stagnant water, and then people will occupationally or recreationally get exposed by coming into contact with this water. So, for instance, in areas where leptospirosis is more common, you have people that have to work and maybe wade through water that has been contaminated with rodent urine that's been infected with leptospirosis. Very commonly, there have been numerous outbreaks of leptospirosis among athletes, especially triathlons. There much more recently was an outbreak in Illinois from triathlon, and triathlons have a swimming portion to it. So what happened is those athletes in the triathlon came in contact with leptospirosis in the lake that the swimming portion of the competition was in. So many individuals came down with leptospirosis, and these types of outbreaks happen all the time. Now, the human is a dead end host. So once humans get leptospirosis and either get treated or clear the infection on their own, that's it. The leptospira is gone. It tends to be a more common infection in males than in females. However, both men and women can get leptospirosis. So here is a morbidity mortality report from the CDC about a triathlon in Wisconsin and Illinois that had occurred, and there was an outbreak of leptospirosis. Diagnosis of leptospirosis usually occurs through serology. That's the more common way that you would diagnose leptospirosis in a clinical laboratory. So the common method is ELISA, or enzyme-linked immunosorbent assay, where you would be looking for IgM antibodies within 1 week after the onset of disease, and the IgM antibodies may persist for months. You can also detect IgG antibodies either 1 or more months after an individual has been infected. Now, since Leptospira is a spirochete, these organisms don't stain with gram stain. So these are not organisms that you're going to put on a slide and do a gram stain and look under the microscope. So that you can't visualize them under a typical bright field microscope. They're only visible through either dark field microscopy, phase contrast microscopy, or immunofluorescent microscopy. So these organisms can be stained with silver stain, and then you would look under a phase contrast microscope. Or you can look under dark field, or you can use fluorescent antibody stains and look at them under a fluorescent scope. Now, Leptospira can be cultured, but it can only be grown in very specialized media. So there's a bunch of specialized media that's been developed over the years for Leptospira. One of these medias are Fletcher's semi-solid media. It's not really a broth, but it's not an agar either. Or Stewart liquid media. Now, these organisms grow at lower temperatures than our typical pathogens that we've talked about thus far that like to grow at body temperature, which is 35 to 37 degrees. Leptospira prefers to grow in the low 30-degree range. So you need very special conditions to grow Leptospira. So Leptospira is not something that you're going to be culturing for in a typical clinical microbiology laboratory in the United States, which is why serology is the more common way to diagnose Leptospira. So you need special media, you need a low temperature incubator, and you would have to let the culture go for at least 1, possibly up to 2 weeks before you would actually detect growth of Leptospira. So after you let the organism grow in your Stewarts or your Fletchers, or there are other medias, you can then look at the organism using dark field microscopy and you can see the cells. So in one of the labs I worked with when I was doing one of my graduate studies, that lab, in addition to the organism I worked on, which was Mycobacterium tuberculosis, we also worked on Leptospira interrogans. So I did use Fletcher semi-solid media and grew up some Leptospira and did a lot of dark field microscopy and electron microscopy of Leptospira. It's a very interesting organism, but it isn't that simple to grow, and it does take quite some time. You can treat infections with penicillin tetracycline. Now, individuals that have renal complications, kidney failure, would have to have hemodialysis done on them. Prevention is extremely difficult because a lot of animals get Leptospira, and then the organism is going to be coming out in their urine. So Leptospira is incredibly difficult to control. If you can control rat populations, that is one way to prevent infections. And you can vaccinate animals to prevent infection in animals. We're now going to move on to the second part of our spirochete lecture.